Microorganisms are tiny living organisms such as bacteria, viruses, and fungi. They are too small to be seen by the naked eye and can be found in large numbers in all kinds of environments: soil, water, air, surfaces, and even our bodies. They colonize the surfaces of our skin, the mouth, nose, ears, and the intestinal tracts. Special precautions need to be taken where there is hair growth and moisture. Most of these microorganisms are considered harmless to us. However, there are other harmful microorganisms that can have negative effects on pharmaceutical products and medical devices. They are pathogenic microorganisms that cause disease. Let's examine the difference between bacteria, viruses, and fungi. Bacteria are single-celled organisms that are classified as prokaryotes. They lack a nucleus and other membrane-bound organelles. They are found in nearly every environment on Earth and can be beneficial as well as harmful to humans. Examples of beneficial bacteria include those found in the human gut and those used in the production of fermented foods and dairy products. Harmful bacteria can cause infections and illnesses. In order to survive and reproduce, bacteria need water. However, they have the ability to survive in very dry environments, which in practice means that they can be dispersed by the flow of the surrounding air. Some bacteria are aerobic, which mean they require oxygen to survive and reproduce. Otherwise, anaerobic bacteria are able to survive and reproduce in the absence of free oxygen. Bacteria are sensitive to heat. Many bacteria do not survive temperatures of 60 degrees Celsius or higher. Other bacteria can form spores, like the Bacillus or Clostridium species. They are very resistant to any kind of environmental conditions and can even survive boiling water. Bacteria are by far the largest and most common contamination hazard in the life science production processes. Fungi are eukaryotic microorganisms. They have a nucleus and other membrane-bound organelles. They can be single-celled or multicellular, and they obtain their food by absorbing nutrients from other organisms. They can be found in many different environments, including soil, water, and air. Some examples of fungi include yeasts, molds, and mushrooms. Fungi can occur in two forms, molds and yeasts. Fungi can be beneficial, as in the case of yeasts used in food production, or harmful, as in the case of molds that can cause respiratory problems. As a fungi fact, molds are the second most commonly found microbiological contaminant in clean rooms. Viruses are non-living particles or infectious agents that require a host to replicate. They are much smaller than bacteria and are made up of genetic material, DNA or RNA, surrounded by a protein coat. They cannot reproduce on their own. They need to infect a host cell, take over its machinery, and use it to make copies of itself. Once inside the host cell, the virus's genetic material takes over the host cell's machinery, using it to produce more viruses. The degree of microbial contamination in pharmaceutical areas can be a hazard depending on the function of the product, the intended use of the product, the type of contamination, and the number of microorganisms present. It's not to be taken lightly, as microbial contamination present in an injectable can even cause death. Sterile products need to be free from viable microorganisms. Even low numbers of microorganisms could pose a potential problem. It is now clear that contaminants in APIs, excipients, medicinal products, medical devices, and other products can come from various sources, such as raw materials, equipment, or the environment. Controlling their levels is essential for ensuring the safety and efficiency of the final drug product. Understanding the origins and nature of microbial contamination are important in terms of seeking and designing appropriate environmental controls for developing effective remediation strategies. And this leads us to contamination control.